Hello, am I audible? Could somebody type it in the chat box? Hello, is this audible to everybody? All right, so your fellow learners are still joining, so we will wait for around 5 minutes, I think, before actually beginning with the session. During which, if in case there is any problem with my audio, you can inform me. Okay, I think I'll begin with today's lecture, and I will not be locking the meeting just in case somebody gets disconnected. They can rejoin again, okay? And it is a fairly long lecture from 3 to 5. So there are a number of things that could be discussed over here. I have prepared, just a moment. I still have a doubt whether or not I am audible, because I have not received any feedbacks as to that.
Yes, ma'am, you are visible and audible.
Okay, all right. Thank you, Sandeep. Okay. And there's no disturbance in the background, right? It is all right if I speak like this.
Yes, ma'am, everything is clear.
Okay. Thank you so much. Yeah, so as I was saying, it is a fairly long lecture. There are about 2 or 3 things that I have prepared. And apart from that, if somebody has certain doubts that are not directly related to the assignments that have not yet been graded, then we can discuss those. These doubts would be the ones pertaining to week one of the lectures, because I'll also be taking 4 classes. So this is week one of my practice session, right? I will also deal with some aspects of week two and the previous year's assignment that was presented for week two, because as I just realized this morning only that you have your assignment for week two also due on 8th, right? So that you are like it helps you guys out. I'll be just a moment. I'll be discussing the previous year's assignments for week one and week two. Apart from that, we will be yeah. Could you repeat? Okay.
Not able to hear anything.
Is it just a problem for Ajay, or is it a problem for the other learners as well?
No, ma'am, it's clear.
Okay, so Ajay, maybe
No, ma'am,
you
it's
could
clear.
connect to a different network because I presume my network is fine. Uh, so you may take this opportunity to connect to a different network. Uh, so yes. I have also gone through certain doubts that were presented by people on the discussion forum, and I have prepared very short slides, and that is just for my remembrance, so that I remember that there was a doubt over there. So maybe we can also discuss upon those topics. Apart from that, if at all you have any queries, you can type it in the chat. We will try to deal it today itself. I might not be very well versed with the entire plethora of doubts that you guys might have. So, in case I'm unable to answer anything, I'll get back to you in the following sessions. So, I hope that how I have planned this arrangement to work is going to suit well with you guys as well. And okay, I think we have waited enough for people, although they are still joining. I'll just wait till 3:10 and maybe then we can start with our the prepared part of my lecture. Okay, so I'll be sharing the screen and then maybe if people are still joining, I'll let them in. Okay. Uh, so I hope you can see my screen. Is it visible?
Yes, ma'am.
No, ma'am.
Okay. Is it visible now?
No, ma'am.
No, no.
No, it is not. Okay. Uh, What do I do for these technical glitches? Just a moment. Is it visible now?
Yes, ma'am.
Okay. Can you also see the slide go forward and backward?
Yes, ma'am.
Okay. All right. Thank you so much. So as I just spoke, I cannot directly discuss this year's assignments with you guys, but I'm sure that some of you might be facing certain questions or certain doubts regarding these assignments. What NPTEL has done is that, okay, so people are still waiting in the lobby. All right. This is going to be a bit of a problem until and unless I log the meeting. People are going to keep joining in. Ah, so NPTEL, for your benefit, what they have done is that they have provided us with the previous year's assignments, and they have allowed us to discuss those particular assignments, and somehow they will be related to your current assignments, or they might deal with the concepts that you are going to use in your current assignments as well. So maybe discussing them might help you, and maybe if not for your assignments, it might help you in your final term exam for this course, right? uh, okay, I see some people are joining again and again. I'm not sure if that is due to some connection issues, but we are going to begin with our first week's assignment for last year. So I would also like input from you guys, and rather than turning on your mics and giving your inputs, I would prefer if they are given on the chat box, because in that way they will be documented and it will not create unnecessary ruckus in the class, right? So, for example, This is the first question. I'm going to read it. I'm going to read the options and I'm going to discuss the options also. And then we'll discuss the answer. But maybe once I'm done with the question, you could all answer in the chat box. And then later we can tally whether our answer is correct or not. And if in case it is incorrect, we'll try to grasp what concept it is based on. Right? So the first question for last year here is the central dogma of molecular biology is an explanation of the flow of genetic information within a biological system based on your knowledge of the processes involved and the cellular compartment where the process take place. Choose the option that identifies the correct pair. So over here, there are processes that are written over here, processes such as replication, transcription, translation, and there are certain cellular compartments that are mentioned over here. They are nucleus and cytoplasm. And you have to basically tell that what is this process actually doing and where this process is happening. So I would first like you all to answer in the chat box. Then we will move on to the solution.
Chat caption is unmuted, madam.
What?
People share views, they are lighted, but chat is blurred. We cannot, we can just
Just a moment.
up and
Okay, okay.
clap and
All
smile and
right, I
wave
get
back.
it. That is why you people were unable to answer earlier. Okay. Okay. Is it possible for you to see them now?
No, madam. Chat is blurred. People
This isn't.
share views, they are lighted. Turn on live caption, don't show chat bubbles. Actually, I think don't show chat bubble.
I have actually enabled it from my view.
Turn off incoming video dial pad. Nothing.
Okay, this will be a problem.
If we can make A as a thumb, B as a clap, C as
Uh, just a moment. Okay, I understand. What I can also do is I can alter it from here. I just need certain permissions to be altered.
share is being blurred. Now people and views, they are open.
Okay. Okay, that is what it says to me also, that it has run into a technical snag.
Madam,
Uh,
A for thumbs up, B for clap, C for smile, and D for raising the hand. If you give that option to everybody, I think it will be okay. We can start. Just like if I want to put D, I will put like this.
yeah, all right. That sounds like a viable option, and maybe that is all we can do right now,
We can
unless
have
and until this is
it will
resolved.
save the time, and next time we can have that chat.
All
Or
right.
if it get corrected, we will start saying ABCD.
Yeah, all right. In the order that they are presenting you with the emotes minus the heart emote, so thumbs up could be for option one, clapping could be for option two, the amused emoji could be for option C, and the surprised one could be for option D, right? Uh, so that is what we are going to do now. Let me, you can still see my screen, right? Okay. So for this first question, I think we have wasted enough time. The answer over here is going to be D, that is translation. Translation is basically a process where RNA is copied into protein, right? So what happens in transcription? It is basically the copying of DNA into RNA. This is correct. But where does it happen? It happens in the nucleus, not the cytoplasm, right? But translation is a process that happens in cytoplasm itself. And so D is the correct option over here. Also, I have put in this visual, which would help you remember which stage basically happens in which cellular compartment. So over here, replication,
You are not
which is
able to see the
basically,
screen again, madam.
okay. Is it visible now?
No, ma'am.
No, ma'am. Sorry, madam. No.
Okay. Whenever I admit a new person, I lose that screen sharing, the successful screen sharing. So maybe during a question, I'm not going to add anybody for the time being. Maybe
Madam,
that will help.
after 15 minutes, don't add anybody, madam. You can do that.
Ah,
15
okay.
minutes.
So I have another pop-up. Now it's visible?
No.
Yes, ma'am. It's visible now.
It's visible now. Okay. Uh, okay. So for a while now, I'm not going to add anybody new in the lobby. Uh, because that basically tampers with the screen sharing. So the answer that we just discussed to the first question is basically the option D, wherein it says that translation, which is basically a process of copying of RNA into protein, takes place in cytoplasm. Thereafter, I was explaining that this particular illustration that I have added in this context, which basically explains which cellular compartment has which process of a central dogma going on. So over here, DNA itself is present in the cell nucleus, right? So replication of the DNA or anything that is directly related to DNA is going to occur in the nucleus itself. So, thus replication as well as transcription, these both activities take place in the cell nucleus. And at what step does it pass out into the cytoplasm? After basically the mRNA processing is also done, right? So during transcription and a pre-mRNA is made. This pre-mRNA needs to undergo certain post-transcriptional steps, right? Post-transcriptional modifications for it to actually become an mRNA. So, what are these post-transcriptional modifications? These are basically the addition of a 5' cap, addition of a 3' poly A tail, and also splicing. Okay, so splicing, as sir has already dealt with in his course itself, is the joining of exons or the coding region and the excision of introns or the non-coding region, right? So after this entire process has taken place, this mature mRNA after the post-transcriptional modifications will move on to the cytoplasm. And in the cytoplasm, translation and post-translational modifications are going to take place. Okay. So this is the next question. A newly synthesized RNA has the following sequence 5' AUC, CUG, GUA, and 3'. Which one of the following option identifies the correct sequence of the transcribed DNA template? So I would like you all to look at the question, try to solve it. Just like we talked about, you can give your reactions for whatever you think is the correct option, and then we'll finally discuss the option.
Sorry, madam, not able to see screen.
Is it a problem with everybody?
I don't know, but it is problem with me.
Yes.
Just see
Yes.
your name, nothing on the
No,
screen.
ma'am.
The
Ma'am,
rest
it
of
is not
you are
visible.
able to see it.
No, ma'am, it is not visible.
Ma'am, just a black screen is visible.
Okay,
There's
all right. I'll try. I'll admit all these people also, and I'll share again.
okay.
Visible?
Yes, ma'am. No, ma'am.
No, ma'am.
I think if it is visible for anybody, it might be a problem for the others. Okay, I am trying to see what the issue is. If in case I'm unable to resolve this issue, I will probably switch the platform on which we are taking these lectures, right? Because then that might be the issue. I'll try to share again, see whether or not you can follow.
Yes, ma'am, visible.
Now,
Visible?
Yes, ma'am, it's visible.
Okay. So try to solve this question. It is a good question. So I'll not give any hints as of now. Okay, can we move on to the solution? I think it's enough time, right? So we'll move on to the solution of this question. And so this is the correct answer. And how it might have been is you might have solved it correctly if you would have paid attention to the 3' and 5' end of the sequence that was presented to you, right? So over here, these are the two things that you need to keep in mind. You always know that transcription is going to go forward in the 5' to 3' direction, right? So if this is the DNA template that they are giving us, this would be reversed. It would be inverted, right? The mRNA that would come out of this would be in, I'm sorry, would be in a 3' to 5' direction. And that is how you had to see this question in order to solve it correctly, right? So accordingly, over here, for A, you get T. For U, you get A. For G, you get C, and so on. So option B and C might have looked confusing, but if this concept was clear in your mind, you would have gotten the right answer. I apologize for the coughing. Moving on to the third question. So it says that an mRNA strand consists of 120 nucleotides. Upon translation, this mRNA strand would form a polypeptide chain that is how many amino acids long? So I also saw that there was a similar question in this year's assignment as well. Only the values I think have been changed. So it directly might help you. It is also a little difficult for me to follow the reactions over here. So I'm arbitrarily taking around 30 seconds in which I hope you will be able to solve the question and then I'll move forward with the answer for the same, right? Okay. So for its solution, we would get around 40 amino acid long polypeptide chain. Why? Because basically mRNA has nucleotides, right? And as most of you might be aware of, that there is a 3-letter code, right? So for every three nucleotides code for one amino acid. So that is basically the 3-letter code that is being followed throughout life on Earth, right? So over here, if we had 120 nucleotides, uh, it would basically code for 120 divided by 3, which would be 40 amino acids. And some of you might have this question that in these 40 amino acids, basically, we are also counting the start codon and the stop codon. So start codon, that is AUG, codes for methionine. That is a universal fact. But stop codon doesn't generally code for anything. So the actual answer should have been 39 instead of 40, because the final 118 to maybe 120, this codon would basically code for the stop codon, right? But since 40 is the closest answer that we get over here, so we are going to choose 40. Over here, what is the assumption is that even the stop codon is basically coding for some amino acid. Okay, let me take a moment to admit other people who are waiting in the lobby, because I have been getting notifications. Okay, I'll share the screen again. Okay, so returning to question number 4 for week 1. Which one of the following mutation types is not likely to generate a truncated protein? I hope everybody is aware about uh, what just happened. Okay, what a truncated protein is, right? So, just in case you are not familiar, truncated protein is basically a protein that is not whole, or that is basically only partially made. Okay, moving on to its solution. Missense protein is the correct answer. Okay, that was a glitch on my part. I'm going to share the screen again, just in case you are not able to see it. Please inform me. Uh, okay, so this is it.
Now able to see, but I think it will go within a minute again.
Ma'am, previous time you was just appeared and like after 2 seconds you just again disappeared.
Now it's
Right now,
right now continuous.
I have actually switched my network as well. I hope it is not going to be a problem now. It's working, right?
Yes, it's working.
Okay. So I don't know whether or not I was able to convey this question or not. So I'll repeat it once again. Which one of the following mutation types is not likely to generate a truncated protein? Right? So, a truncated protein is basically a protein that is not fully formed. It is basically half a polypeptide or something that has stopped midway, right? So the correct answer for this was basically missense mutation. So over here, what they have asked is what is not likely to generate a truncated protein, right? So a truncated protein will be formed if there is a nonsense mutation. Why? Because what happens in a nonsense mutation is basically that there is a stop codon that is encountered and this protein will stop forming at whatever place it is encountered, right? So the full protein will not be able to be formed. The other option over here was frame shift mutation. So what will happen in a frame shift mutation is that there will maybe be an insertion or a deletion due to which basically this reading frame will be altered, right? So it might happen that when this reading frame is altered, basically, it basically leads to the a code that codes for a stop codon again. Similar with a point mutation as well. If a point mutation such as an insertion takes place or this U is basically deleted or a transition or translation takes place that this is basically changed to a G, right? Again, it will code for a stop codon and the truncated protein will be formed. But such a thing cannot happen in case of a missense mutation. Why? Because missense basically means that uh, the codon or the mRNA was coding for one particular amino acid, and now instead of that amino acid, it is coding for a separate amino acid, right? But it never says that it is going to code for a stop codon. Otherwise, it will be termed as a nonsense mutation and not a missense mutation. Thus, a missense mutation will form a full protein only some place in that protein, one amino acid might be altered. Okay, which is the case for sickle cell anemia, for example. So, moving forward to question number 5. Given below is a karyotype of a patient suffering from a congenital hernia. Examine and identify the options that list the correct karyotype and cause for the defect. So for ease of understanding, they have also placed an arrow as to where the defect is. You have to basically identify what this defect or this karyotype will be named. So again, I'll take an arbitrary 30 seconds maybe. Also, it might not take that long to do this question. So, can you think of what basically this chromosome 15 or the chromosome that has a defect actually looks like? Or if you have studied that in your course for week 1. It is a practical karyotype example of something that you have studied. Okay, moving forward towards basically solution. This will be a 46XY deletion, right? So first of all, you have to look at the sex chromosomes and you have to be very particular as to what you are choosing, XX or XY. It might have been possible that you might have been in a situation such as an exam or a test where you are in a hurry to solve questions, and this might have been your first option and you might have ticked it. Okay, so this is something that has to be paid attention to, and it is clearly visible that in this chromosome number 15, one of the homologous chromosomes is much smaller than its other homologue. So it has bound to be a deletion, right? Uh, if it would have been a translocation, you would also have seen some other chromosome involved over here. There might have been another arrow pointing out to another chromosome. So that is not the case. This is a 46XY deletion. And in this particular chromosome, uh, the deletion leads to formation of a ring chromosome, right? So this is a 15q26 deletion, which leads to formation of ring chromosome 15. So just in case you are not aware about the terminology, uh, I don't know if I'll be able to draw or not. Yeah. So if this is a chromosome, right? Let's say this is chromosome 15. So what is this terminology P and Q? So if this is the centromere, the short arm of the chromosome is known as the P arm or the petite arm, and the long arm of the chromosome is known as the Q arm, right? So 15th chromosome Q arm has uh, Q arm locus number 26, might be somewhere over here, right? So 26 is basically the locus number. There might have been a deletion somewhere over here, due to which it basically forms a ring-like structure that you have already studied in your lessons also. So moving forward, I hope I'm audible. Do I need to check?
You are
Okay, I guess I'm
audible and
audible.
screen sharing was also good. Why you stop?
Yeah, so I'll join again. It should not happen that I am blabbering and I might have been disconnected, right? So that is why I need to check once in a while. Okay, I'll share again. Okay. So this is the next question. Which one of the following option correctly matches the syndrome to their respective karyotype? So you have the options in front of you. Maybe we can switch to the solution slide in 10 seconds. Yeah. Somebody's trying to say something.
Clean filter C, madam.
Oh, yeah, we are going to see the solution. So it is indeed Klinefelter syndrome, and these are certain syndromes that you have already studied: Down syndrome, Turner's, and Klinefelter in your lecture. Apart from these there are certain other ones that you have studied also, and this Patau and Edwards commonly are asked in competitive exams, and sometimes even I have seen in previous assignments for this course they have been asked. So I have just included this table so that you are aware about these trisomies as well. So trisomy 13, that is Petau's syndrome. Trisomy 18, which is Edwards syndrome. Trisomy 21, which is one of the most common, Down syndrome. Then Klinefelter syndrome, it is not important that it is always double XY. It might also be triple XY. So as like the more the defect, if it is 48 instead of 47 also, the symptoms or the condition might be even worse for the patient, right? Then there's Turner syndrome, which is monosomy of X. Crido-Cat, which has also been discussed, is a deletion in the P arm of chromosome 5. I hope now you are aware of what P and Q arm are. And Prader-Willi syndrome and Angelman syndrome, which have been studied or taught in the context of imprinting disorders and uniparental disomy. So they involve deletion in chromosome 15. Then the next question is an allele, sorry, such a tongue twister. An allele is a mutant version of a gene, a variant of a gene that may not cause a disease, one of the several variants of a gene present in normal population, or all of these.
D.
Okay, so indeed it is all of these, and it is just a definition. So I hope it does not require any further explanation. I hope you all are aware of what an allele is. Uh, Basically, the alternate forms of a gene, as you might have studied in your textbooks. So moving forward to question number 8. Identify the options that correctly fill in the phenomena described in the statement given below. The Philadelphia chromosome that causes chronic myeloid leukemia is an abnormally short chromosome 22 and it is a product of a dash event. What is
A.
a kind of a chromosomal aberration that takes place in the formation of Philadelphia chromosome? Also think about what other chromosome is involved in this, right? Because
D.
okay,
Translocation.
yeah, D. So basically, translocation is going to take place, and the chromosomes that are involved over here are chromosome 9 and chromosome 22. Sorry. So these chromosomes respectively have two genes, gene regions. One is BCR and one is ABL. You can read further about it. It is not that important over here. So translocation within these chromosomes leads to a longer chromosome 9, as you can see in this karyotype also, a longer chromosome 9 and a shorter chromosome 22. And this shorter chromosome 22 is basically known as the Philadelphia chromosome. And it is basically one of the major causes of chronic myeloid leukemia, right? It is a type of cancer. Okay. So the second last question for this particular week assignment is the unessential components are excised off of the from the pre-mRNA strand with the help of splicing factors to generate the mature mRNA. Identify the component which is not a part of the mature mRNA strand that has to be translated. So which out of these
A.
is not a part of the mature mRNA
Intron.
strand? Yeah, so for everybody's reference, the options are intron, exon, 5-UTR, and poly A tail. So the solution is intron. So over here, uh, the mature or the final spliced mRNA is still going to contain the 5' and the 3' UTR. It is still going to contain the poly A tail. It is also going to have the 5' cap along with it and the exons that are basically spliced together, right? But what is it going to lack? These introns. Okay. Okay. So the final question for this particular assignment is the region of the chromosome that joins a pair of sister chromatids is known as the dash, and the chromosome ends are referred to as the dash.
Ma'am, D.
D.
Yeah.
Centromere
Okay.
telomere.
So that is right. Centromere, I have not put an illustration, so I am just going to again. So this part that is going to attach the two arms is known as centromere, and these end regions are known as telomeres. Okay. So for this part, like this ends my first presentation. I have a couple of more for you guys. And thank you for sitting through the first one. I will admit more people who are waiting in the lobby. Just a moment. Yeah. Let me put it into the presentation mode so that there is no problem with that later.
Excuse me.
Okay, so I can actually ask you guys, we have enough time. I think we'll be able to cover both the other parts also. So what do you want me to do first? I have the previous year's assignment for week 2, which I have just prepared this morning because I realized that you have your submission date for a week 2 assignment as well. So I thought it might help you guys. And the other thing that I was originally planning to do was that just touching upon certain topics that were taken up in the first week's time. And there were certain things that I thought need to be discussed in that week's lectures. And there were certain queries that I picked up from the discussion forum. So we can discuss that part as well. So what do you want to do first?
Ma'am, the discussion part.
The discussion part. Okay. So, okay, now that the chat box is actually not working, I have enabled the Q&A. In case you have any you can try it out and tell me whether it's working or not. If in case you have any questions, you can post it in the Q&A. I'll try to address those questions in either in today's class, or I could maybe get back to you regarding those Q&As in the following classes also. Uh, okay, a couple of more things that I think I'll forget later. Uh, you are not able to access the meeting, but are you able to at least see what I am typing in the chat box? Did you get my message? Am I audible?
You are audible,
No,
madam, but you are not
no,
able to
ma'am.
see what you are typing.
Okay, okay. So you are not able to see that. I just wanted to like
Madam, sorry
I
to
don't,
interrupt. Just
as
one
such,
thing
have a platform where I can directly interact with you guys, and it will help me if you could put in your queries to me so that I can prepare my next class accordingly, and it would help you guys also better. So for that, maybe you can, maybe I can type it over here itself. You can see my screen, right? Oh, sorry, I have not shared my screen.
not now.
Uh, hold
Madam,
on.
just one query. Most of the syndromes they are diagnosed by physical characters. Question
Uh,
number 6
uh,
said, okay. But just
yeah.
interesting thing I'm asking you. Clean printers, it was in published in some 1968 report. Most of the
Yeah.
juvenile delinquents were found out to be Klinefelter. So Klinefelter syndrome, they look like a common man. That's what my query is. Means they are not diagnosed by characteristics, just like Patau, Edward, Downs, Turners. They can be very well easily with dysmorphic features or some
Yeah.
characteristics are
Okay.
there.
So
They can be easily diagnosed, but that's not case with I think Klinefelter.
yeah, so why that might be is because like in Klinefelter, there are two X chromosomes present, and along with that, there is additional Y present. So if there will be a Y chromosome present, it will definitely turn out to be a male, right? And the only symptomatic changes that were happening in this male was due to the extra X or overexpression of the X chromosome, right? So this X chromosome, you guys might be aware of the dosage compensation, right? So anyway, in most of the cells, are you aware of dosage compensation?
Yes, madam. Another
Wherein
X chromosome is
females
a bar
have
body.
two,
Is turned
yeah.
out into bar body. Another X chromosome in female, it turns out to be bar body. Means it is coiled in the cell.
Yeah, so there is this natural phenomena through which if there is an extra X, it is going to randomly get inactivated, right? So in most of the cells, that is what is going to happen. And there might not be a presentation or a symptom for that disease. But at some points, it might actually interfere with the general being of that individual. That is why the uh, like the symptoms that are seen in in the individuals with Klinefelters are not that extreme, and that is why even triple XY phenotype genotype is also seen, right? Otherwise, I don't think in case of any other you would see something that is more than a trisomy, right? But in case of this particular sex chromosome, something that is basically quadruple is also seen, because again, that much tolerance is present in the body to counter it, right? So there are only issues with certain uh, fertility factors in the Turner syndrome and the Klinefelter syndrome patients. That is also because by default, I am actually also going to discuss this with respect to SOX9, that by default, the human body, like the embryo, when it is developing, by default, it develops into a female. Right. So if there are not those Y chromosome factors that intervene and tell it to become a testes or tell it to basically develop into a male, the default pathway taken by the embryonic development is towards a female only. So again, that is, so how did we start this? I got off topic. Okay, yeah. So we started as to why these conditions are basically not apparent in younger individuals, right? Because the sex chromosomes are not that anyway, not that developed in the sexual features are not that developed in the younger individuals, and it is not going to hamper with your quality of life. It is just going to hamper with your how do you say the procreation process, right? So that is why it is not that significant. So what I was going to do was I'll share my screen. I was actually typing. So you can see my screen and you can see where my cursor is, right?
No, ma'am.
Okay, not able to see my screen. Let me try again, okay? Now?
Yes, ma'am.
Okay. So I have added my email ID over here. I have also highlighted it. Maybe you can note this down because I have no other means of communication because the chat box over here is not working. So maybe you can note this down. Try not to spam my email ID, okay? But maybe you can send in your queries and doubts and what you want to be discussed in your upcoming lectures for the next 3 weeks that I'm going to take lectures for you guys. You can basically ask or maybe just intimate me what you want to know more about pertaining to your course, right? So, okay, I'm going to stop sharing this particular screen. I hope everybody has my email ID now. It is arveenk21@iitk.ac.in. Okay, and we'll be moving forward with our next discussion part, right? Okay. Okay. So is this slide visible as well?
Yes,
Yes.
ma'am.
Okay. Uh, So it is a very brief, there were not very difficult concepts in week one, right? In the upcoming weeks, the concepts keep getting deeper and deeper. So whatever I could fetch out or whatever problems I thought I could solve, I have listed them over here. We will try to solve them one by one. So this is just it. Okay, before beginning. It is a combination of basically questions that I have come up with, concepts that I wanted to sort out, and revision from this week, right? So there is somebody waiting in the lobby. Let me just, before I start, let me admit these people also. Okay, still visible?
Yes, madam.
Okay, all right. So, the major discussion point for week one lectures was central dogma. So, central dogma is basically this flow of information in which basically DNA, the flow of information, which is unidirectional because it occurs according to the classical view as to when central dogma was presented. It was presented as a genetic flow of information which is unidirectional, that is, it moves in one direction only from DNA to RNA to protein. Also, there was another hypothesis that came up during the classic understanding of central dogma, which was basically that one gene codes for one polypeptide, right? So these were discussed in the previous lectures. Then, who actually presented central dogma of molecular biology? It was Francis Crick. What else did Francis Crick do, or what else is he credited with? It is basically the structure of DNA, right? So Watson
Double
and Crick,
standard.
they also gave the structure of DNA as we are aware of today. Along with that, Francis Crick in 1957 also presented central dogma of molecular biology during one of his lectures. Uh, then I wanted to discuss what this one gene, one enzyme hypothesis was and who actually proposed it. So, Sir also discussed this. It was proposed by George Beadle and Edward Tatum, who also later received the Nobel Prize for physiology or medicine in 1980 for 1958 for this particular hypothesis. Although later this hypothesis proved to be incorrect, which we will discuss why. Uh, so what they actually said in this was that there is one gene which basically codes for one mRNA, which leads to uh, uh, one enzyme. So the protein or the polypeptide was not the term that was used over here. Over here, it said that genes code for polypeptides, which are basically enzymes, right? So they are involved somehow in the metabolism or the metabolic process of life. So what was the organism in which basically Beadle and Tatum did their study? So you guys have studied this in Sir's lecture. Anybody would like to contribute?
Ma'am, neurospora.
Yeah, so it was Neurospora. So in that, they basically identified that there were certain precursors which basically led to and in the presence of this first enzyme, it led to formation of ornithine, which in the presence of second enzyme led to the preparation of citrulline, which in turn finally formed arginine in the presence of enzyme 3. And what they found out was that in case any one of these gene was defective, the enzyme that was formed due to that gene was also defective, and the for these sorry ornithine, citrulline, and arginine could not be formed in case such a defect occurred. So, in case gene 2 was defective, so enzyme 2 would not be formed, and the precursors would be able to form ornithine, but this will not be able to form citrulline or arginine, right? Because this process itself is hampered. Similarly, if there was a defect in gene 1, enzyme 1 would not be formed, and even ornithine would not have been able to form, right? So that is what they studied, and that is how they proposed one gene, one enzyme hypothesis. But how is this hypothesis incorrect? That is something that we need to understand. So, this hypothesis, like I just told you, states that the major function of the gene is to form an enzyme, right? But it is not only an enzyme that is formed. There are other proteins that are formed. These proteins could be maybe collagen or elastin, or they could be receptors, cell surface receptors, or they could be basically pathogen recognition particles. All these are proteins which have different roles, right? So, genes do not only form enzymes, they form polypeptides, like they code for or they carry the information to code for polypeptides in general. These polypeptides could be hormones, or they could be enzymes, or they could be collagen, elastin, dystrophin, structural proteins, surface receptors, anything, right? So that is one place where this hypothesis was incorrect. The other place was that one gene could also code for more than one enzyme, right? So this we have now understood. Now it is a universal fact that one gene through alternative splicing could have different splice products, and thus it could have different polypeptides that it codes for, right? So there is not just one gene and one product. One gene with multiple mRNAs could code for multiple polypeptides. How is it possible? Due to alternative splicing, right? This is also why, like, we have so many proteins, so many cell surface receptors, and so many enzymes that are prepared in our body intrinsically. And the number of genes that are present in our body are only 30,000, right? There are even more number of pathogen receptors than that. So how is that possible? Is one of like one of the phenomena that makes it possible is due to alternative splicing only. The other phenomena is when it comes to those surface receptors, it comes under the domain of immunology, that VDJ recombination that we will not touch upon right now. Thereafter, let us also refresh our modern view of central dogma. So the modern view states that not just DNA, but RNA and proteins can also self-replicate. So if in case you get a question that which of the following biomolecules has self-replication capabilities or self-replication ability, and you get options as DNA and RNA, DNA, RNA, and protein, only DNA and protein. So you know what option to pick? DNA, RNA, protein, all of them can self-replicate, which was not what Francis Crick thought, but this is actually what is true. Okay. Just a moment, I'll admit people from the lobby. I will also have a sip of water. Okay. Tell me if the screen is still visible.
Yes, madam.
Okay. So the next view is reverse transcription. So the reverse transcription is one of the phenomena that when it was founded, most contradicted central dogma, right? So what is reverse transcription? What is transcription? Transcription is basically the passage of information or the copying of information from DNA to RNA, right? So what will reverse transcription be? It will basically be the reverse of transcription. That is the passage of information from RNA to DNA, right? So this reverse transcription, it is widely used in molecular biology as a technique also, and it was actually brought upon as a natural phenomena. It was found in certain RNA viruses. So certain RNA viruses, what they do is that they infect the host. They inject their RNA into the host. Then they synthesize, they do reverse transcription. They basically convert uh, this RNA information into a DNA information, and then this DNA could then be integrated into the host DNA, and it basically helps in the viral replication. So that is how viruses basically use reverse transcription, and that is how this concept was first studied, and later it was also used as a molecular biology technique. Next is viruses without DNA. So, as I just mentioned, there are RNA viruses, but not all RNA viruses need to or have the ability to do reverse transcription also. Okay, there are certain viruses that will directly just translate from RNA to the protein that has to be formed, right? So DNA will not be involved at all in this process. So this also defies central dogma because DNA is anyway not present in these viruses, right? Or involved in the flow of information. Then non-coding RNAs and ribozymes. So non-coding RNAs. We are going to discuss about what non-coding RNAs are, I think in the upcoming slide or in a couple of slides. So these RNAs basically do not code for proteins, but they are present. DNA basically codes for certain coding RNAs and certain non-coding RNAs. The coding RNAs will basically code for proteins and polypeptides, while the non-coding RNAs, they will just remain as RNAs and they will perform other functions and not code for proteins as such. And what are ribozymes? So ribozymes are basically another fascinating examples of these uh, RNA particles that basically defy central dogma, because in this basically the RNA itself is going to behave as a uh, enzyme or a protein, right? So it is going to perform the function of a protein by behaving as an enzyme itself and not translating into a protein. So, yeah, non-coding RNAs, like I was talking. So, what are non-coding RNAs? I feel that not everybody is very well aware of what non-coding RNAs are. It is not just the intronic regions or anything, right? Non-coding RNAs are basically RNAs that perform functions other than coding for proteins. So we are also going to study what certain non-coding RNAs are, some of which you all might be very well aware of. But it is just that sometimes people are not aware about the terminology, right? That, okay, tRNA is a non-coding RNA. Okay, I was not aware of that. Okay, so just to clear things out, I have made this illustration also, which I have adapted from Biorat. So you can go onto their website and you can also read more about it. So non-coding RNAs basically are coded by RNA genes. So genes that code for these RNAs are known as RNA genes. Again, I have another illustration to explain this. These non-coding RNAs could be either regulatory non-coding RNAs or housekeeping non-coding RNAs. Now, this is something that we have also basically studied or read about in terms of DNA, right? Housekeeping genes and regulatory genes. So, whatever has housekeeping in it basically means that it is very essential for the functioning of over here, maybe a cell, or for the human body itself, right? It is essential for functioning. And regulatory basically means that it helps in the regulation or certain maybe upstream or downstream regulation of certain processes. So some examples of non-coding housekeeping non-coding RNAs include tRNA, rRNA, snRNA, and snORNA. Right, while regulatory non-coding RNAs could be divided into small non-coding RNAs or long non-coding RNAs. I forgot to mention the size range for this. Small non-coding RNA is below 200 nucleotide of length, and long non-coding RNA is more than 200 nucleotide of length, right? So small non-coding RNAs include piRNA, miRNA, siRNA, and scRNA. Long non-coding RNAs had complex names, and I do not think you should go into such complexities as of now. But these RNAs that I have listed over here, I have also given their size range and functions in this table. It is for your reference. So tRNA is the transfer RNA. rRNA is the ribosomal RNA. We are all aware of these two RNAs at least, right? So they are involved in the protein synthesis machinery. snRNA is a small nuclear RNA. It basically helps in the RNA processing and intron splicing. SNORNA is a small nucleolar RNA which helps in rRNA processing. And piRNA, miRNA, and siRNA are basically RNAs that are involved in RNA interference or RNAi, right? I have mentioned it over here as RNAi. These are RNA interference. pvRNA is also involved in regulation of transposable elements. siRNA and miRNA are very widely used in molecular biology again these days for CRISPR. So these are basically involved in RNA interference, which is another very interesting phenomena, and I'm sure you would love to read about it. Okay. And I also just talked about RNA genes, right? So just to clarify what RNA genes are, is basically that suppose this is a DNA, right? This is a DNA strand, and the boxes over here, the blue boxes, they represent genes. This first gene, let me just number these genes, gene 1, gene 2, and gene 3. So this first gene, it basically codes an mRNA which codes for a protein, right? So this is a normal protein coding gene. But these other two genes, that is gene 2 and gene 3, they do not code for a protein, right? There is no protein that is present over here, but they code for RNAs, right? Regulatory RNA and tRNA. So these code for non-coding RNAs. So these genes will be referred to as RNA genes. Okay. So this is another question. That does an exon contain only coding region? This was asked and answered in one of the lectures itself by Ganesh sir, right? So, but I felt that this is a point that people miss and they slip. It slips out of their mind. So, the question over here is, does an exon contain only coding region? So, to answer it,
No,
let me, yes, somebody wants to answer.
no, it contains
What else
contains
does it contain?
other things like non-coding RNAs.
No. Okay. So that is why it was a very crucial thing that Sir said, but I also think that a lot of people miss this. So that is why I wanted to emphasize on this point in this particular slide. I will explain over here. Again, there are people waiting in the lobby, so let me just admit them. Okay, I will have to keep time also in my head. It is around 4:30. I will try to finish everything that I have prepared. Okay. So does an exon contain only coding region? So the answer would be no. And how will we explain this answer? That we have to see it now, right? So this first illustration, it basically has this. You can consider this as mm, a pre-mRNA, right? So this is a pre-mRNA, and this is exon 1, this is exon 2, and this is exon 3, right? And the regions over here, this region is an intron, this region is an intron, right? So the final mRNA will not contain this region. It will also not contain this region. These will be spliced together, right? And so we will have, okay. So we will have this exon 1, exon 2, and exon 3. But along with this, which is the coding part, these exons are basically the this part, and this part are the coding part, the ones that are present in green. But apart from this, there is also this 5 dash UTR and this 3 dash UTR, right? So this is a part of this exon only. This is a part of this exon only, the final exon. And these are the non-coding parts, right? So that is why the exons will not just contain the coding region, they will also contain the untranslated region, right? Introns or non-coding RNA is not a part of it, but the 5 dash untranslated region and the 3 dash untranslated region are a part of these exons. And so it is not just coding region. This is the same illustration that we also used in discussion of one of the questions earlier. So again, the final spliced mRNA has, along with these exons, the 5 UTR and the 3 UTR, which are parts of this exon only. Apart from this, there is also poly A tail and 5 gap. Okay. All right. Then this is another important thing that I feel that you guys should know now that uh, uh, one of the lectures also talked about spliceosome, right? So there is also a rule associated with spliceosomes or with splicing, which is basically the GUAG rule, right? So, what I have titled the slide is how does the spliceosome recognize introns? So, I think it is a very intriguing question. How basically it will know that the exon ends over here and the intron starts and the next exon starts over here, right? So, there is one rule that explains this, which is the GUAG rule. So, this says that the first two and the last two dinucleotides of an intron are GU and AG respectively. And so, these snRNPs in the spliceosome, they basically recognize these GU and AG dinucleotides and basically excise the introns out of the pre-mRNA as splicing the other two exons. So the region of 5 GU is attached to the region of 3, the region 5 of GU. So means the region upstream of GU is attached to the region downstream of AG, right? And the intronic region from the middle is removed like this. So this is how basically splicing occurs, and this is how spliceosomes recognize introns. I hope I'm not going too fast, right? These are just some small facts that I had to give you. There's not much explanation that is required for them. Okay. This slide I have only kept because I don't know whether it was a genuine doubt or not, but in the discussion forum, there was somebody who asked, explain genetic code, right? I don't know whether the person is attending the class or not, but I thought that, okay, I will take out 5 minutes to try and explain what a genetic code is. So genetic code is basically a 3-letter code, right? And these 3 letters are basically formed from 4 alphabets. These alphabets over here, they are A, T, G, and C. But when we talk about RNA or protein, like A, T, G, and C in DNA, right? But when we reach at an RNA or a protein level, T gets replaced by U or uracil. And so these 4 letters are G, U, A, and C, which are present at the center of this table, right? So these 4 alphabets basically come in combinations of 3, right? So 3-letter codes are formed by these 4 words, which basically code for some or the other amino acid. or stop codon. So there are in all 64 uh, codes, 64 three-letter codes, because there are basically four letters raised to power 3, which is the number of, which is the size of the code, which basically leads to 64, right? Out of these 64, 61 code for amino acids and 3 code for stop codons. Okay. I'm so sorry. So these three that code for stop codon could be found over here. For example, this. So UGA codes for stop codon, UAA codes for stop codon, and UAG also codes for stop codon. These three code for stop codon. Apart from these, there is one start codon which also codes for methionine, which is basically AUG, which codes for methionine, as well as it is also the start codon. And the other amino acid codes can also be found over here. For example, for leucine, there are four codes that are present. So there is a redundancy in the coding for leucine. For example, CUU, CUC, CUA, CUG, all code for leucine, right? But there might be some like methionine for which only one codes. For example, tryptophan. So UGG codes for tryptophan. It is the only codon that is going to code for tryptophan. So this is how the three-letter code basically works. This particular circle is the key for this code. And okay. I think we have already discussed the chromosomal abnormalities also. So I'm going to skip that part and I'm done with this slide officially. Moving on to the difference between mutation and variation. So there was one thing that Sir mentioned in his lecture that mutation could basically affect the survival of an individual, whereas variation does not affect the fitness or the survival of the individual. So that is one thing that you have to keep in mind. Apart from this, mutation is a permanent alteration of a nucleotide base in an individual, whereas variation is referred to difference between individuals or groups of individuals within a species, right? Then what I have just discussed, the same thing came across again. Then mutation is basically, since it is a alteration in the nucleotide, it will be the result of a mutagen, which could be radiation or chemicals. But variation could arise due to genetic recombination also. For example, the cross that occurs in prophase 1 that could lead to variation in individuals. Genetic drift could lead to variation in individuals. Even the epigenetic changes or the environmental changes could also lead to variation in individuals. Then mutation is always genetic, whereas variation could be either genetic or environmental, or it could range between them, and it could be epigenetic, right? Okay. So this is again from the discussion forum. Somebody wanted to study about lactose intolerance. So lactose uh, is basically a protein that is present in milk or dairy products, right? Which is very essential for a developing human being because the primary source of food for them is milk, right? So what basically uh, happens is that there is a enzyme lactase that is present in the that is secreted from the walls of small intestine. I'm so sorry. I have triggered Thank you, sir. Okay. Um, So there is this enzyme lactase that is secreted from the walls of small intestine, which basically breaks down lactose, which is a complex carbohydrate, into it is basically a disaccharide, not a complex carbohydrate. It is a disaccharide. It breaks it down to glucose and galactose, which are then used by the body, right? And the gene responsible for the production of this enzyme is basically LCT gene, which is present on chromosome 2, right? So its chromosomal location over here I mentioned is at as 2q21, which again, now I think you might be aware that it is on the long arm of chromosome 2 and present on locus 21, right? So this is its chromosomal location of this gene. So what generally happens, or what generally used to happen earlier, was that in the normal adults, it was a general condition that over time this gene will lose its function. The lactase will not be secreted in the small intestine, and lactose will not be broken down to glucose and galactose. And this basically means that the person will become lactose intolerant, that is in the absence of lactase. And if the person is lactose intolerant, the lactose will now move down to large intestine. And if it moves down to the large intestine, bacterial fermentation of this lactose will take place, and it will cause some unpleasant effects such as gas, acids, and flatulence, which could sometimes even lead to abdominal pain due to the presence of gas, right? So, this was a general condition, but now there have been polymorphisms due to which, in general, populations have become lactose persistent. So, we don't need lactase in our life, but in general, due to natural selection, you might say, the polymorphisms that are prevalent now are basically those of lactose persistent people, right? So now you don't generally see people of maybe my age who are lactose intolerant that frequently as you might have uh, maybe 3 or 4 decades earlier, right?
Madam, I don't think so
Yeah.
because same doubt arose me in Ganesh sir was taking lecture. Since
Yeah.
long ago, every human is taking a milk or milk product.
Yeah.
So lactase is
Yeah.
present, lactose is present only in milk, or it is also present in the milk product. Because we are drinking tea from not from 4 decades, from long ago. From
Yeah.
Dwapar Yuga or from Sri Krishna era also, they are using consistently milk. There were everywhere there is milk supply, and in everything means even Ashwathama Mahabharat mentions he wanted to drink milk because the peoples from rich families they were drinking the milk. That story is always told. So
Yeah. So,
this is all down
like
to
you,
there was
hmm,
some period in which there was no lactates in a human being.
no, there was no such age
Eh,
where
not
there
not
was no,
not in the older age group there was no lactate. That is means possibility is less. That's what I want to ask.
yeah.
Because
So
every
it
age,
also depends
every
from population to population. Now, this is a concept that it is a generalized concept that we are studying. So for example, I'm not aware of the exact percentage, but if I were to take an arbitrary number, I would say that maybe 4 decades ago, the worldwide population had 40% of the people who were lactose persistent and 60% who were lactose intolerant by the time they turned 30. Okay, it is an arbitrary number. So we are talking about world population over here. But eventually, like India, for example, you might not have come across lactose intolerance in your grandparents also, I think, that often, right? But if you talk to somebody who is presently settled in Sweden, or maybe somebody who is present in a Caucasian country, or maybe Switzerland, Sweden, Norway, some such nation, right, where lactose intolerance is like very persistent. So if you ask them about whether or not they can digest lactose, they probably won't be able to, right? It also depends from population to population. Also, it was never such that lactose was never present in the adult population, right? Polymorphisms means that this allele was also present, but the a number of people who did contain such alleles, which would make them lactose persistent, were much less, right? But now, in general, we are talking about the world population right now also. But now, in general, people from maybe India who had lactose persistent population spread to US, right? Which had lactose intolerant population. People procreated over there. Now, their progeny is much more likely to still be lactose persistent, right? Unless and until there is some environmental factor that is making them lactose intolerant, these populations have the polymorphisms or the alleles which would let them tolerate lactose throughout their life also, or maybe up till they are very old, maybe 70, 80, right? So that is what Saur meant. Am I clear? Like, was I able to solve your doubt?
Thanks.
If in case you still have a doubt, I'll try to explain it again. Polymorphisms
No,
you all
madam.
are aware of, no?
Okay. I will go again
Polymorphisms are
into
basically.
comparison and see if I have doubt. I will again let you know, madam.
All
Oh,
right. All right.
thank you.
Also, I want to clear it out whether everybody is aware of what polymorphisms are. So polymorphisms are basically, uh, for example, like we talk about alleles. Alleles are alternate forms of a gene. So polymorphisms are different types of alleles that might be persistent in a population. One individual can have only two alleles. But in all, a population can have multiple alleles or multiple possibilities, right? For example, blood group. So in that, A is one possibility, B is one possibility, AB is another, and O is another, right? But one individual can have only two alleles. It could be AA, AO, AB, something like that, right? But overall, all four possibilities basically exist in the entire population. So such different forms of alleles are basically known as polymorphisms. Okay. Also, one of the lectures, Kozak sequence was mentioned. So I thought of refreshing your memory as to what Kozak sequence is and what is a similar sequence that is present in prokaryotes. I should have laid this slide up and I should have asked it from you guys, but I forgot. So first, let us discuss what Kozak sequence is. So it is basically a consensus sequence that is present in eukaryotes. It contains the start codon within it, and it is the region where translation basically begins, right? And this is the sequence. And where is the start codon in this? This AUG, right? So this is present in eukaryotes. We have also studied a similar sequence that was used to be present in prokaryotes, right? What was this sequence? It was Shine-Dalgarno sequence. I think people who have studied molecular biology in their graduation, post graduation, or are going to study molecular biology might be aware of this, right? So we study transcription, translation, replication in prokaryotes, then transcription, translation, replication in eukaryotes. Then we study where Scheindel-Carnot in prokaryotes, Kozak in eukaryotes. These things might be ringing a bell for some people, and it might be new for some people. So what this Scheindel-Carnot sequence is basically this sequence, right? It is located in prokaryotes. It is located 8 bases upstream of start codon. So unlike Kozak sequence, which itself contained the start codon, this is present 8 bases upstream of start codon. So if this is the start codon, it will be present over here, right? And it is also the site where ribosome binds. So why will ribosome bind? Because translation has to begin, right? So both these are for signaling translation to begin, and they have different names, and they are present in different types of um, animals, eukaryotes and prokaryotes, for example. Okay, moving forward, ring chromosomes. So we just saw an example also of what ring chromosome was, how the chromosome 15 deletion in that particular case led to ring chromosome. So these are basically circular chromosome structures that form when a chromosome breaks from two ends and the broken ends are fused together. So the exact mechanism of how this happens is still elusive. So we are not very well aware of that. But it there are two methods in which this ring could be formed. One is basically telomere dysfunction, that the telomeres, there will be some problem with them, and they will basically just roll and form a circle and fuse. Right, so the entire chromosome is present in this ring chromosome. And it could also be a result of double-stranded breaks. In this, what will happen? Suppose there is a break over here and over here, the telomeres will be lost, and the rest of the chromosome will basically have those staggered sticky ends, right? So they will just roll up and they will form a ring. And why will we lose these telomeres? Because basically they cannot be replicated, or they cannot be uh, yeah, they cannot be replicated. What is one of the primary prerequisites of replication is basically having a centromere, because the spindle needs to be attached somewhere, right? And these have lost the centromere, so they are anyway of no use and they will be lost and degraded. Then uniparental disomy. So this was another concept. Let me just check the time. Okay. Uh, Holding this off a while, I would like to mention that maybe we'll not be able to do the week 2 assignment today, right? So I'll just finish this particular segment, because if I hurry up, it will be of no use for you guys also. We can anyway proceed with that and another discussion setup in the following week. Okay, so uniparental disomy. Uh, Uniparental disomy, we can break this word down, and what it will tell us is that there is one parent, right? There is uniparental, just a moment. Okay, I had to admit somebody in the meeting. Ajay, do you want to ask a question? Her hand is raised. Okay. So moving back to uniparental disomy. So uni means a single, right, parent. Uniparental, single parent. Disomy, that means that two chromosomes will be coming from a single parent. That is basically referred to as uniparental disomy. So there are different ways with which this could happen. How is it possible is that the first way would be, suppose this we are going to take all the left circles as the egg and all the right circles as the sperm. All right. So in this case, we say that the egg basically has two chromosomes, but there is a nullisomy in the sperm, right? So the resultant child will basically contain two chromosomes, but both of them will be coming from the egg because the sperm was null. It had no chromosome, right? So this individual will be completely all right because it still has two copies of chromosome. But that is a different case that both of these copies arose in the egg only, right? Another possible case could be that there is a normal sperm and again there is a disomic egg. So it will form an individual with three chromosomes. Now, such individuals will not be tolerated. Like there will be some or the other uh, rectifying or proofreading mechanism which will go on, and probably if this still persists to be three chromosomes in this individual, then it will probably be removed or degraded, right? So maybe the rectifying mechanism comes and it knocks out one chromosome, right? One of these three chromosomes is knocked out. So probability wise, since these are two and this is just one, there is a high probability that this chromosome will be knocked off. And again, the two chromosome containing individual that we now obtain again has these two chromosomes from the same parent only, right? It is very well possible. So this is another basically path through which uniparental disomy could originate. The third one could be that the egg could be normal, but again, the sperm could be having a nullisomy, right? So there will be no chromosome that will be contributed by the sperm, but there will be one chromosome that is being contributed by the egg, right? This individual now has one chromosome and there will be certain rectifying mechanisms again that will be working over here. So in order for it to still be qualified as as a complete individual, maybe this gets copied and into another chromosome, and this is the individual that we obtained, which again obtained both its chromosome from the same parent because the sperm did not contribute into this. So this is how uniparental disomy could basically originate, right, in different formats.
Ma'am,
So there, hello, yeah.
ma'am, is the uniparental disomy the chromosomes that were being considered? Are these only autosomes?
Yeah, so the example, actually, I'm not sure about this because all the examples that I have ever come across are all autosomes. Also, it will not always be tolerated. So that is why there are only a few examples that you will come across. Because, for example, for trisomy or monosomy also, you come across very few examples because such instances occur very rarely where certain such huge defects are actually tolerated. So I am not very well aware of it, but I would say that yes, the examples I have come across are all autosomes. I'll check and I'll inform you.
Okay, ma'am. Thank you.
Madam, but it is theoretical possibility only, or does that's what why I was asking. Ki examples are there because how it can be just autosomes? Sex chromosomes will also be there. No? Sperms don't contain any chromosome, so it will be also sex chromosome. It will be transferred. No chromosome in the sperm, then it will be transferred as a for survival. the baby will require XX chromosome, or it will be only all these will be females only. Only chance
No,
is that.
so this is just an example in which we were taking this particular as an egg
Are we
and this
considering
particular as an
all
huh.
the living material or just for human beings? This uniparental disomy, we are considering
Actually,
human beings or in all living
we are
beings.
considering this in the case of human beings over here because there are a lot of other, for example, in plants, aneuploidies, even euploidies are
That
tolerated.
is possible. That's what I am
Yeah.
asking. That is
Over
possible,
here,
but
also, I'm not sure who asked that question because I was not on the screen where I could see the viewers. But in a sense, you can also say that Klinefelter is a uniparental disomy. I'm not sure about it. I'll have to check. But if you think about it in a theoretical way, the double X,
madam, clean printer also have Y, no? How? Achha, disomy means all the autosomes are from mother and only Y chromosome is from the father.
no, in case of Klinefelter, there was XXY, right? It
Yeah,
could have been that double X was from the mother and Y was from
mother.
the. We are considering all the autosomes were from different parents. And there was double X from the mother and Y from the father, or it
Okay.
could also have been that there was X from the mother and XY from the father.
Yes, madam.
Although I'm
There.
not very sure whether we can call it
Uh,
uniparental disomy or not, but theoretically seeing, this is
that
what
is
it
the
means.
sex chromosome. Okay, correct, madam. Sorry to interrupt. That is sex chromosome uniparental disomy, na? Klinefelter is. If X
Hmm.
is from mother and Y from the father, and X is from
Yeah.
mother and Y from the father. And in that case, we don't know whether all autosomes are also from father or mother.
It is very highly unlikely that all chromosomes have a uniparental disomy. It will be the case for one of the chromosome, maybe,
One of the chromosome.
because
That's what.
otherwise it will be a condition called euploidy, right? That the
Euploid.
ploidy of the chromosomes itself is increased if all chromosomes are in multiples, which is not tolerated in humans.
Okay, so but for the query which was asked before me, you said you just seen it in an autosome. But the example you are giving is now for the sex
That
chromosome.
is what I'm saying, that I am not sure whether or not it is a case of uniparental disomy. If we theoretically see, I'm not sure about it. That is what I first said. Because
Yeah.
Klinefelter could, like theoretically speaking, this is what is happening in Klinefelter also. Right? But
I
I
think,
have never personally read it as uniparental disomy.
but if the case is such, the Klinefelter is a practical example of uniparental disomy in sex chromosome.
I'll have to check that.
Okay, madam, please. Thank you.
Yeah. Um, okay. So these were two imprinting disorders that were discussed, Angelman and Prader-Willi syndrome. In these particular disorders, what was talked about was that, for example, there are two chromosomes. One is a paternal and the other is a maternal chromosome, right? In the paternal chromosome, the Prader-Willi gene is active, means it is functioning. So there will be no Prader-Willi disorder in this particular chromosome. But the Angelman gene is imprinted. So imprinted gene over here means that this could lead to Angelman syndrome or the disorder, right? While in case of maternal chromosome, what happened was that the Prader-Willi gene was imprinted, means this disorder could occur, but the Angelman gene was active, right? So this disorder will not occur, right? It is a normal gene. When these are present together, we have one copy of active Prader-Willi and one copy of active Angelman. So that is why there will be no disorder, neither Angelman disorder nor Prader-Willi syndrome. No syndrome will occur, right? But if in case there is a deletion of the parental chromosome, right? So we just have the maternal chromosome now. This maternal chromosome has normal functioning Angelman gene, but it does not have normal functioning Prader-Willi gene, right? So this will basically lead to Prader-Willi syndrome. Why did the syndrome occur? Because the maternal chromosome did not have a properly functioning Prader-Willi gene. And because of certain maybe chromosomal aberration, there was a deletion of the paternal chromosome, and only the mother or the maternal chromosome got a say in whether or not these genes will work, right? So whatever gene was not functioning caused a syndrome. So that was simply what imprinting disorder was. And I have also basically these two genes are present on chromosome 15. There are certain regions that are marked for these Prader-Willi syndrome region and Angelman syndrome region. We do not need to get into this much detail as to what exact locus they are on, but they lie between Q11 to Q13. 15Q11 to 15Q13, just in case it is asked anywhere. Okay. So SOX9 gene. Let me admit people in the lobby. How come people are joining this late? You had another class at 5, right?
Yes, ma'am.
Have you received the what, like the login credentials for those
Leave for
classes?
this 3 to 5, not for 5 to onwards.
5 to 7, you have not received the login
Link.
credentials.
Yes, madam.
Okay, my colleague is actually supposed to take that class. That is why I am asking if you have the credentials for that class. I don't want to hog her time as well. Okay, so and I am just seeing that it is 5-7. I would like your consensus as to whether I should move forward, like I should finish this portion, or should I do it in the following class?
Please finish, madam.
People might be busy. Is it okay for everybody if I finish?
Yes, madam, because that
Yes, ma'am.
class
Because
also
we
will
have
take
not
Yes,
time.
ma'am.
received the link also.
Oh, all right. All right. So this part I'll finish. Then we'll see what is to be done in the next class. Okay, people are waiting in the lobby.
Hello.
All right. Okay. So SOX9 gene. This was mentioned in one of the alternative splicing maybe defects, right? So I found this as one of the interesting topics to inform you guys about. So SOX9 basically stands, and there was also some discussion regarding it in the discussion forum. So just to clarify it, SOX9 stands for SRY box transcription factor 9 gene, right? What is SRY? SRY is basically sex determining region Y, which is present on the Y chromosome, right? It will basically specify whether it will, because it is a Y chromosome, it will specify that the individual has to basically develop like a male, right? So testes need to be developed. Ovarian development has to be stopped. These are the things that SRY will basically signal. And it also uses this gene SOX9, which is present on uh, chromosome 17. Sorry. Oh, this is my last slide. Good. So on the long arm of chromosome 17, it is present, and it also SRY basically signals SOX9 to start testicular development. So there are two major functions of this gene. One is the determination of sex, by basically turning on the testicular genes, and the other is basically embryonic skeleton formation. So that is also where SOX9 plays a role. Also, mutations in SOX9 are associated with male to female sex reversal, right? So, if in case this SOX9 gene is a let us first see in this representation how SOX9 is basically going to work. So, there is this SRY gene that I just told you about. It is present on the Y chromosome. What it will do is that it will turn off the ovarian genes. So, in the earlier discussion, while we were discussing the question answers, I told you that the default pathway of development of a human is basically as a female. So, if it will not receive signals, any other signals, every embryo will develop into a female, right? So, there is a very small time, particular time that is set wherein signals for testicular development should be received. Otherwise, the default development towards the female takes place. In general, that is hello. I hope I'm back on.
Yes, ma'am.
Okay.
Madam, it's
Huh.
continuing. Please
Okay.
continue.
So, what this SRY will do is that it will turn off these ovarian genes and it will turn on the testicular genes, right? So, testes formation signal should go. Also, what it will do is that it will turn on the SOX9 gene, which is present on chromosome 17. Then SOX9 also has certain functions like keeping these ovarian genes turned off, also keeping these testicular genes turned on, right? This SOX9, along with some other factors, one of which is SOX8. So SOX8 and SOX9 have a redundant function. So these will basically determine the fate of the individual as a male by first promoting testicular development and then sustaining testicular development, okay, until this development is complete. So that is it. These were certain things that I wanted to discuss, and thank you in a genetic kind of way. Uh, Thanks for your patience. Let me stop presenting. I
Oh,
hope that you have all received my email ID. Try not to spam my email. I will not be able to read them if there is a lot of spamming going on. You can just inform me what all topics, apart from the week two assignment that is to be discussed, what all topics do you want me to discuss next week? Okay. So maybe I'll also come a little bit more prepared.
Yeah, could you please show the questions you just solved before throughout this session?
Uh, do you want me to share them with you? Like,
Yeah, the
if the
questions.
chat would actually work, then I would share them also. But the chat is not work. Do you just want me to go through those slides once?
Yeah, yeah, yeah. Just in a like in a few glance.
Okay, just a moment.
No, chat is not working. It's inactive only. Thanks, madam, for your time and nice explanation. Thanks
Yeah,
from all
you
of
Okay.
can
us.
just yeah, you can run through the slide.
Thank you so much. Those who are done can leave. Thank you for attending the talk also. And Ajay, I think, asked me for the questions, if I'm not wrong. You are the
Yes,
one who is on top of my screen somehow. So I am displaying the questions for you, then I'll log out and the others can leave. I'm not going to teach anything new, just here for the questions, right? I'll show the these slides, answer slides. So this is question one. Moving
Ma'am?
on to question. Hanji, yeah.
Yes, as you mentioned that the SRY gene is located on the Y chromosome, so it's
Yeah.
active for males, and for females there is no Y chromosome. So the random process
SRY
that
located on
did not.
70.
I'm sorry, somebody interrupted that question.
So for females there
Yeah.
is no
Sorry.
Y chromosome, so there is
Yeah.
no ovarian blockage occurs during the ovarian formation.
Ah, yeah. So, the Y chromosome is not there. So the SOX is not involved as such. But there are other factors that still need to be involved. So some of them are like R-spondin 1 or beta catenin or WNT signaling. So these signaling pathways still go on if the ovarian development has to take place. So the signaling pathway is different. SRY and SOX are not involved, but yes, we still have uh, like control over that pathway also. So control for that is also present.
Ma'am, is there
Even though
also
the
the
default
Mullerian duct?
pathways,
The
even though the
Mullerian duct
default pathway
inhibition.
is female. Yeah. What?
Also, the Mullerian duct inhibited in males for the Wolffian duct.
The which one was in females? Uh, I get confused in this.
Females,
Just a moment. Do you mind
Mullerian
me
duct is
checking
for females
once?
and Wolffian duct is for males, I guess.
Uh, just let me check. Okay. I am actually looking for it and I'm unable to find it.
Okay, ma'am, you will elaborate in the next class about this.
I will.
That will be okay. Thank you, ma'am. Okay.
Thank you.
Yeah, could you please run through the questions?
Okay. Question number 9, the second last one, and the last one. All right.
Now, the all 10 questions. No,
Yeah.
don't explain, just need to see the questions asked.
Can you still see my screen?
No, like we can't.
Oh,
Yes,
you
madam, I can.
can't. I thought my screen was visible. I was doing it all through my conversation with the I'm not sure who I was having the conversation with, but I was changing my screen actually. I thought it was visible. Wait. Huh. So this is question 1.
Yes, ma'am.
Shall I move forward?
Yeah.
Next
Just
question.
Okay. So this is 2.
about 10 seconds to each questions, and it will be done within minutes or so. 1 minute or so.
Okay. And 3.
Yeah. Yes. Yes, ma'am. Go forward. Yeah. Yes, ma'am. Yeah. Yep.
Okay.
Yeah.
This is the final one.
Yeah. Thanks for sharing all the questions.
Welcome.
Thanks.
All right. So guys, I'll also be leaving the meeting. Let me just stop the recording.